Hi everyone! Today I'm diving into the fascinating world of stem cells, the building blocks of regenerative medicine. Stem cells have a unique ability to differentiate into specific cell types needed for growth or tissue repair. Think of them as blank slates, ready to become any cell the body requires. This transformation is guided by both internal and external signals within the body. Internally, genetic factors play a crucial role, while externally, environmental cues and chemical signals direct their development. Now, let's talk about their self-renewal capability. This is what allows stem cells to divide and reproduce themselves over long periods, unlike regular cells that wear out over time. Stem cells can keep dividing, ensuring a constant supply of fresh cells. This self-renewal is essential for tissue regeneration and repair. Imagine a scenario where an injury occurs. Stem cells rush to the site, differentiate into the needed cell types, and then start repairing the tissue. This incredible process is the cornerstone of regenerative medicine, offering hope for treating a variety of conditions, from heart disease to spinal cord injuries. By understanding the mechanisms of stem cell action, researchers like you can unlock new possibilities for healing and regeneration. The future of medicine lies in harnessing the power of these remarkable cells.
All right, let's dive into the fascinating world of stem cells. Stem cells are the foundation of regenerative medicine and hold the potential to revolutionize how we treat a myriad of diseases. First up, we have embryonic stem cells. These powerhouses are derived from the inner cell mass of a blastocyst, an early-stage embryo. They are pluripotent, meaning they can differentiate into any cell type in the body. This versatility makes them incredibly valuable for research and potential therapies. Next, let's talk about adult stem cells, also known as somatic stem cells. These are found in various tissues throughout the body, like bone marrow and fat. Unlike embryonic stem cells, adult stem cells are multipotent. This means they're generally limited to differentiating into cell types of their tissue of origin. Despite this limitation, they play a crucial role in maintaining and repairing the tissue they reside in. Moving on to induced pluripotent stem cells, or iPSCs. These are adult cells that have been reprogrammed back into an embryonic stem cell-like state. As iPSCs are pluripotent, they have the same potential as embryonic stem cells to differentiate into any cell type. This breakthrough technology provides a significant advantage as it bypasses the ethical concerns associated with using embryonic stem cells. Finally, let's discuss perinatal stem cells. These are obtained from sources like amniotic fluid and umbilical cord blood. Perinatal stem cells are also pluripotent, offering a versatile resource for regenerative medicine. Their accessibility and potential make them an exciting area of research. In summary, stem cells come in various forms, each with unique characteristics and potential applications. From the versatile embryonic and perinatal stem cells to the specialized adult stem cells and revolutionary iPSCs, the future of stem cell research is incredibly promising.
Stem cells. They are the body's raw materials, the foundation for every organ and tissue in our bodies. But what makes them so special? Let's dive in. Stem cells have two remarkable abilities. First, they can differentiate. That means they can transform into various types of cells, muscle cells, red blood cells, neurons, you name it. Second, they can self-renew continuously, producing more stem cells. This makes them an internal repair system constantly working to replace damaged or worn-out cells. There are several types of stem cells. Embryonic stem cells come from early-stage embryos and can become any type of cell in the body. They're pluripotent. Then we have adult stem cells found in tissues like the bone marrow and brain. These are multipotent, meaning they can develop into a limited range of cell types. Now let's talk about their clinical uses. One of the most well-known applications is in bone marrow transplants. Here, hematopoietic stem cells, which can develop into all types of blood cells, are used to treat conditions like leukemia and lymphoma. Stem cells are also being explored in regenerative medicine. For example, scientists are investigating how stem cells can repair damaged heart tissue after a heart attack or how they can regenerate neurons in spinal cord injuries. The potential is vast. Finally, induced pluripotent stem cells. These are adult cells that have been genetically reprogrammed to an embryonic stem cell-like state. They hold promise for personalized medicine since they can be derived from the patient's own cells. Stem cells represent an exciting frontier in medical science. Their ability to repair and regenerate tissue opens up numerous possibilities for treating diseases and understanding human development.
Let's talk about something groundbreaking in regenerative medicine-induced pluripotent stem cells, or iPSCs. iPSCs are a type of pluripotent stem cell generated directly from adult cells. Imagine taking a skin cell from a patient and reprogramming it to become a versatile stem cell. This technique was first pioneered by Shinya Yamanaka in 2006, and it's been a game changer ever since. So, how are these iPSCs created? It starts with adult cells, like skin or blood cells. Scientists introduce specific genes into these cells, usually using viral vectors. These genes reprogram the cells, turning back their biological clock, making them behave like embryonic stem cells. Voila, you have iPSCs. Now, what makes iPSCs so special? They have the remarkable ability to differentiate into almost any cell type in the body. This means they can become neurons, heart cells, liver cells, you name it. They hold the key to personalized medicine. In regenerative medicine, iPSCs offer tremendous potential. Since they can be derived from the patient's own cells, they greatly reduce the risk of immune rejection. This opens the door to patient-specific therapies. For instance, if a patient has a damaged heart, iPSCs could be used to generate healthy heart cells tailored to that individual's genetic makeup. The applications of iPSCs don't stop there. They're also used for disease modeling and drug testing. Researchers can create iPSCs from patients with specific diseases, allowing them to study those diseases in a dish and test potential treatments in a controlled environment. However, there are challenges. The reprogramming process can sometimes introduce genetic mutations, and ensuring the safety and stability of iPSCs in clinical applications is crucial. But the potential benefits far outweigh the hurdles. In summary, iPSCs are revolutionizing regenerative medicine by providing a source of patient-specific cells for personalized therapies. They hold promise for treating a variety of conditions, from heart disease to neurological disorders. The future of medicine is looking bright with iPSCs at the helm.
These cells hold incredible promise for regenerative medicine, and it all starts with reprogramming adult somatic cells. So, what exactly are iPSCs? In simple terms, they're adult cells, like skin or blood cells, that have been reverted to an embryonic-like pluripotent state. This means they can potentially develop into any cell type in the body, just like embryonic stem cells. The magic happens through a process known as cellular reprogramming. Here, we introduce a specific set of genes, often called the Yamanaka factors. These factors are OCT3/4, SOX2, KLF4, and c-MYC. Named after Shinya Yamanaka, who first discovered them in 2006, these genes essentially reset the developmental clock of the adult cells. Now let's break down the role of these Yamanaka factors. OCT3/4 and SOX2 are critical for maintaining the undifferentiated state of cells, acting as master regulators. KLF4 helps in the self-renewal and proliferation of the cells, while c-MYC enhances the efficiency of the reprogramming process. When these factors are introduced into adult cells, they work together to erase the cell's current identity and revert them to a pluripotent state. Imagine taking a specialized cell, like a skin cell, and wiping the slate clean so it can start over with the potential to become any type of cell. This breakthrough not only avoids the ethical issues associated with embryonic stem cells, but also opens up new avenues for personalized medicine. By using a patient's own cells, iPSCs can be generated that are genetically matched, reducing the risk of immune rejection in therapies. In essence, iPSCs are revolutionizing our approach to medicine, offering new hope for treating various diseases and injuries.
Induced pluripotent stem cells, or iPSCs, are revolutionizing the future of medicine. Imagine taking a skin cell and reprogramming it back into a pluripotent state, capable of becoming almost any cell type in the body. This breakthrough technology is opening up endless possibilities in research and clinical applications. One of the most exciting uses of iPSCs is in disease modeling. By creating specific disease models in vitro, researchers can study the disease mechanisms with unprecedented detail. For example, iPSCs derived from patients with genetic disorders allow scientists to observe disease progression and test potential treatments in a controlled environment. iPSCs are also playing a pivotal role in drug development and testing, and they provide a reliable platform to assess drug safety and efficacy in a patient-specific manner. This means that drugs can be tested on cells derived from the patient, potentially reducing the risk of adverse reactions when the drug is administered. This method is a game changer for personalized medicine. In regenerative medicine, iPSCs hold promise for tissue repair and replacement. Imagine generating pancreatic beta cells to treat diabetes, neurons to address neurodegenerative diseases, or cardiac cells to repair heart conditions. These advancements could transform how we approach treatment and recovery, offering hope for conditions previously thought to be untreatable. Personalized medicine is another area where iPSCs shine. Since iPSCs are derived from the patient's own cells, treatments developed using these cells have a reduced risk of immune rejection. This means therapies can be tailored to the individual, making them more effective and safer. The potential of iPSCs is vast, and their impact on medicine is just beginning to unfold. As research progresses, we can look forward to a future where treatments are more personalized, effective, and accessible. Thanks for watching. Stay curious and keep exploring the incredible world of iPSCs and their applications in modern medicine.